Hi everyone, welcome back to Useful Genetics. This is Module 3. We're going to talk about proteins first, and then we're going to talk about the phenotypic consequences of proteins. So we'll start by thinking about what proteins are, polymers of amino acids, whose order is specified by the bases in the messenger RNA, themselves derived from the genes. Proteins then, once they've been synthesized, they fold into complex but very specific, distinct structures that are driven by interactions between the different parts of the protein and determine the kinds of functions that the protein can carry out. So, proteins are polymers. You remember this term from Module 1, where we used it to describe DNA and RNA. Proteins are also long chains of subunits held together by a common kind of backbone, but like DNA and RNA, protein subunits can have different side chains, analogous to the different bases of DNA. All of the subunits of proteins are called amino acids because they have an amino group on one end, and you'll see it, the chemical structure there, and a carboxylic acid group on their other end. There's the carboxylic acid of this amino acid. There's another amino acid. Now, the amino acids are held together by bonds called peptide bonds, and they're formed when the carboxyl group of one amino acid forms a bond with the amino group of an adjacent amino acid, like that. And there's your peptide bond. So this happens sequentially, starting at one end, so there's an end terminus that's free, and then all the subunits are assembled. Now this assembly with the free end terminus on one end and a C carboxyl terminus on the other end, this assembly happens in the ribosome, and it's carefully driven by this very complex molecular machine. You remember I showed you a molecular structure picture of the ribosome in Module 2, and it was an extraordinarily complex molecule. Now, each amino acid has different chemical structure between its N and C ends. This is called its side chain. Here are examples of specific side chains. They can be very simple. Glycine's side chain is just a hydrogen atom, or a little more complicated, or moderately complicated. At their most complicated, amino acid side chains are smaller and less complicated than the bases of DNA. But it's these side chains that give proteins their very complex properties. Because there are many more different side chains, there are 20 different amino acids that can be assembled into proteins. Proteins, of course, are much, much longer than 20, but it's like the English language. We have 26 letters, and we can make an almost infinite variety of words. The same with proteins, with 20 different subunits to choose from. Very, very wide range of biochemical properties are possible. The order of the side chains, the order of the amino acids that are assembled into the protein, is specified by the messenger RNA, ultimately by the DNA. This is actually a DNA sequence shown here. And it's read in the 3 base codons that we described earlier. So here's one codon, the next codon. Each codon specifies a particular amino acid with a particular side chain. Now, these amino acids were chosen by natural selection. Natural selection acting on the DNA, acting on the phenotype of the organism, favoring organisms whose DNA specified proteins with sequences of amino acids that carried out particular functions that benefited the organism. That's the fundamental principle of how genes evolve. So natural selection selects for protein sequences that cause the protein to fold up in a way, because proteins are long chains, but they fold up into compact structures. They fold up in a way that they have particular biochemical properties. Each kind of protein having a different structure, different kinds of biochemical properties because of its different sequence of amino acids. And its activity and the folding are caused by the interactions of the different amino acids with each other within the peptide chain. So if there's the peptide chain, it will fold up because different parts of it interact in specific ways. And its activity is also influenced by the other molecules in the cell. Now, what we've done, we've talked about how amino acids form peptide bonds, which are the backbones of proteins. Proteins have not only a peptide bond backbone, but they have side chains. Every amino acid of the 20 different kinds has a slightly different molecular structure between its N and C bonds. And these complex structures give proteins their properties. The sequence of the amino acids, which determines the properties of the protein, is in turn specified by the sequence of the messenger RNA, which is transcribed from the sequence of the gene. And this amino acid sequence determines the structure and the function of the protein. Coming up next, we'll talk about protein folding, about how proteins fold, and we'll introduce the different kinds of functions that proteins can have. I hope to see you there.